A really interesting paper was recently published in the Journal of General Physiology, which looked at the ways that poisonous vertebrate animals can tolerate their own poisons. This is a really interesting question that scientists have speculated about for years. Many animals, like several frog species, produce extremely potent poisons that are capable of seriously injuring or even killing an adult human, oftentimes with enough poison left over to kill even more people. The big mystery is, if these tiny little frogs and other poisonous animals are producing chemicals that are extremely dangerous, so much so that they can kill, large mammals like an adult human, how are they able to tolerate their own poisons? How can they tolerate this stuff? Why doesn't it kill them? There's some kind of evolutionary explanation behind some sort of mechanism here, but that's the question. What is that mechanism? That's what this recently published study sought to explore, and it's the topic of today's Science News. So let's begin with the introduction section of the paper. It's packed with details that provide context and background information, and it sets the stage for their experiments and their findings. In the introduction, they say, quote, "Many organisms harbor various small molecule toxins that target ion channels as a means of defense from predation. Among these, batrachotoxin, BTX, a diet-acquired steroidal amine found in distantly related vertebrate lineages, including poisonous birds and neotropical poison frogs, stands out because of its lethality and its unusual ability to facilitate opening and prevent inactivation of voltage-gated sodium channels. This lipophilic steroidal neurotoxin is thought to bind in the voltage-gated sodium channel's inner pore." Unquote. Okay, so to summarize this, they're talking about small toxic molecules that can disrupt ion channels. And if you've listened to Series 2, ion channels are super important for cells, because they're one of the primary mechanisms that cells use to maintain various ion gradients and internal homeostasis. And one of these small toxic chemicals is the super dangerous BTX, or batrachotoxin, which they suspect binds to the inside of sodium channels. As sodium channels are fundamental to the mechanisms and actions and processes of neurons, this BTX, this batrachotoxin, can mess up nerve signaling in the affected tissues, usually in muscles, which leads to paralysis. So, continuing with the introduction, the researchers say, quote, "How vertebrates that bear BTX or other small molecule toxins avoid auto-intoxication remains unresolved. Toxin-resistant mutants of target ion channels in host organisms or their predators have been suggested as the primary drivers of toxin resistance, an idea supported by examples of detrodotoxin, TTX-resistant, and saxitoxin, STX-resistant, voltage-gated sodium channels, as well as epibadidine-resistant nicotinic acetylcholine receptors found in toxin-carrying metazoans." Unquote. Alright, so what they're saying here is that it's still unknown how these animals, like these poisonous frogs and birds that produce batrachotoxin, it's still unknown how they survive their own poisons. Most of the proposed explanations and models involve receptors that have mutated such that they are resistant to their poisons. This idea makes a lot of sense, and as they said, for some poisons, like detrodotoxin and saxitoxin, there's evidence of specific sodium channels that do have mutations that do make them more resistant. Even in some poisonous frogs that produce epibadidine toxin, they have mutated nicotinic acetylcholine receptors that are resistant to that. But what about BTX? What about these batrachotoxins? The researchers continue, saying, quote, "In poison frogs, a voltage-gated sodium channel domain 4 segment 6 pore-forming helix mutation has been proposed as the BTX resistance mechanism." Just a quick aside, the mutation has a really long name. They call it DIV-S6N-T to simplify the direction of the mutation. But as I'm reading this, just for simplicity's sake, if it comes up again, I'm just going to call it DIVS6. Just so you know, that's the name of the mutation. So continuing, they say, quote, "Although the DIV6 change reduces BTX sensitivity when tested in rat sodium channels, this variant occurs with very low frequency among Phyllobates terribilis, and is absent from Phyllobates arotenia, another poison frog having high BTX levels. Given these issues and the absence of functional studies of poison frog voltage-gated sodium channels, Whether BTX-bearing animals rely on voltage-gated sodium channel mutations or other BTX autoresistance mechanisms remains unclear, unquote. So here they're saying the proposed mutation that confers batrachotoxin resistance works in rats. So in this model organism, they can get it to work. But it doesn't really exist in several poison frog species that naturally produce batrachotoxin. They don't have that mutation. So that means that this proposed mechanism may work, in a theoretical, experimental context, but it may also be incorrect because we don't know exactly how it works in the actual wild poison frogs. Consider what the study co-author Dr. Daniel Miner Jr. said in a press statement, quote, "There haven't been any functional studies of poison frog or Petui sodium channels," so that's a type of poison bird. "So whether batrachotoxin-bearing animals rely on changes within their sodium channels or alternative resistance mechanisms remains unclear," unquote. Now in the rest of the paper's summary, the scientists explained that they tested this genetic mutation, this DIV-S6 sodium channel allele, on some species of Central and South American poison frogs, specifically Phyllobates terribilis and Dendrobates tinctorius. And they also tested it on a poison bird from New Guinea, Ptohui uropygialis meridionalis, or PUM. But in all of these poison animals, these two poison frogs and this poison bird species, they found that this DIVS-S6 mutation actually had a deleterious effect on the animals. So this supposedly protective channel variant, this protective mutation that works in rats, quote, "compromises channel function and fails to produce BTX-resistant channels when tested in the context of poison frog sodium channels," unquote. So that's weird. This mutated channel that was proposed as an explanatory mechanism for batrachotoxin resistance actually doesn't work at all in these batrachotoxin-producing frogs and birds. They also tested saxitoxin, or STX, which is another small molecule toxin found in unmodified poison frogs. The researchers found that the frogs were resistant to this poison too, and yet the poison frogs had sodium channels that are, quote, "sensitive to both toxins," unquote. So what is going on here? This is really confusing. So what is going on here? The frogs in the Ptuhui bird produce a poison that could kill them 100 times over. Their sodium channels are demonstrably vulnerable to it, and the supposedly protective mutated channel was actually harmful in the frogs. So how are they doing it? How are they resisting the batrachotoxin? With further research, it turns out that quote, saxaphilin, SXPH, a high-affinity STX binding protein found in frog plasma and frog organs, can protect and rescue voltage-gated sodium channel expressing cells from STX poisoning by sequestering the toxin, unquote. So basically what they're saying here is that the frogs contain a powerful protein that exists in their blood plasma and in their organ tissue. This protein, called saxaphilin, or SXPH for short, is known to be able to bind with saxitoxin, and it can help pufferfish, for example, stop from poisoning themselves. The researchers found that saxaphilin can also bind strongly to batrachotoxin. So in effect, saxaphilin, this protein in the blood plasma and the organs, is like a poison sponge, or like a chemical dog catcher, rounding up loose BTX and STX molecules. That's wild, and it's not at all what was predicted by the proposed explanation involving a sodium channel with protective mutations. The less popular toxin sequestration theory turns out to be right in this case. The toxins are absorbed and sequestered by this saxaphilin sponge. It's able to outcompete the sodium channels as a binding receptor for the toxins, so the toxins are all picked up by this molecule before they can bind to and disrupt the function of the sodium channels. The scientists say, quote, "Our data challenge the hypothesis that BTX autoresistance is rooted in voltage-gated sodium channel mutations, underscore the trade-offs between toxin-resistant mutations and fitness cost, and highlight the potential importance of alternative mechanisms, such as toxin sequestration, as strategies for protecting toxin-bearing species from autointoxication and environmental small molecule threats," unquote. Dr. Daniel Miner Jr., the co-author I quoted earlier, was also describing the value of this research in understanding the various evolutionary forces that created the poisons and the mechanisms that protect against them. Dr. Miner Jr. said, quote, "These strategies might not only offer a general means of toxin protection, but could also act in pathways involved in safely transporting and concentrating toxins in key defensive organs such as the skin," unquote. He also said, quote, "Understanding these pathways may lead to the discovery of antidotes against various toxic agents," unquote. So there you go. A super cool study and really fascinating findings that include elements of biochemistry, cellular biology, zoology, and evolution that even has some potential medical value too.
Hey, bud.